Hi, this is Dr. Freddy. Today I'm going to discuss about the cell cycle. Where a cell arises, there must be a previous cell. Just as plants arise from plants and animals from animals. This is called the cell doctrine, which was proposed by the German pathologist Rudolf Virchow in 1858, carried a profound message for the continuity of life. Cells are generated from cells, and the only way to make more cells is by division of those that already exist. All living organisms, from the unicellular to the multicellular mammals, are products of repeated growth and division. If you look at a human being, we are composed of about 30 to 100 trillion cells, and each of these cells vary in size, which can be ranging from 15 to 25 microns in size. And each of the cell has a nucleus which measures about 1 to 2 microns. And inside this nucleus we have 46 chromosomes, which contains the genetic material in 3 billion nucleotides in one strand of DNA. This DNA contains the genetic material, which is made up of 4 little molecules called as the A, T, G and C. This makes up life around us. Well, let's talk about the cell cycle. A cell reproduces by performing an orderly sequence of events in which it duplicates its contents and then divides into two. This cycle of duplication is called as the cell cycle. The eukaryotic cells have evolved a complex network of regulatory proteins known as the cell cycle control system that governs progression through the cell cycle. The most basic function of the cell cycle is to duplicate accurately the vast amount of DNA in the chromosomes and then segregate the copies precisely into two genetically identical daughter cells. The processes define two major phases of the cell cycle. One is called as DNA replication, which occurs during the S phase. S is for synthesis, which requires about 10 to 12 hours. After the S phase, chromosome segregation and cell division occurs during the M phase, which takes about 1 hour. Most cell requires more time to grow and double their mass of proteins and organelles inside the cell. So to allow more time for growth, extra gap phases are inserted into most cell cycles, which is called as the G1 phase between the M and the S phase, and G2 between S and M phase. Thus the cell cycle is divided into G1, S, G2 and M. G1, S and G2 together are called as the interphase. A human proliferating cell in interphase might occupy 23 hours and 1 hour for the M phase. Now let's look at the proteins that are involved in the cell cycle. For this we require a set of proteins called as cyclins and cyclin dependent kinases. Each of the phases of the cell cycle requires a different cyclin and a different cyclin dependent kinase for the cell cycle to progress. Let's look at each phase of the cell cycle and describe the cyclins and the cyclin dependent kinases that are required for the cell cycle to progress. If the cell finds out that the conditions are not favorable for division, it can go into resting phase, which is called as a G0 phase. Each of these cell cycle phases have their own checkpoints whereby they can check whether the progression of the cell cycle is okay and then go forward. So these are called as cell cycle checkpoints. Now let's look at the G1 and the S phase transition. For G1 S phase transition, you need a cyclin D and a cyclin dependent kinase, which is cyclin dependent kinase 4 or 6. So we need cyclin D and a cyclin dependent kinase 4 or 6. These two have to be together to function. So this is blocked by coming together by another protein which is called as P16. So if this P16 expression is reduced, then cyclin D and CDK4 can combine together and become functional. So in the absence of P16 and other inhibitors which block CDK4, these combine together and become functional, which in turn phosphorylates RB. Once RB, that is retinoblastoma, is phosphorylated, it can combine with another transcription factor which is called as E2F, which will allow the G1 to S transition to happen. Once this job is done, this cyclin D, CDK4, RB, E2F should dissociate. For that to happen, a protein should appear which is P16. The presence of P16 will block CDK4 combining with it. So if P16 blocks cyclin D and CDK4 is not in combination with cyclin D, it cannot phosphorylate RB. If RB is not phosphorylated, E2F will dissociate. If this happens, then this progression or the function of this set of cyclins and cyclin dependent kinase is done. So the next set of cyclins will take over, which will appear during the S phase, that is cyclin E and CDK2. This will make sure that the DNA replication happens in a precise manner. If there is any mistake during this phase, the cell cycle will stop, arrest the cell cycle, check for the mistakes, rectify it by way of DNA repair, and then progress. In case the cell evades this mechanism of checkpoints that are here during the G1 phase or the S phase or any phase of the cell cycle, it can go ahead with the mistake and the cell becomes abnormal. So this is the starting point of cancer. A small mistake which cannot be arrested by the cell cycle, or the cell tries to evade the checkpoint mechanism that is there inside the cell, it becomes an aberrant cell and it will go through the cell division and it will become from one cell to two different cells, and a clonal expansion will happen, which is the basis for cancer, which I'll talk about in the next video. Now for G2 to progress, you need another set of cyclins which is called as cyclin A and CDK2, and to check whether these are going on properly, another set of cyclins, that is cyclin A and CDK1. Now during the M phase, the M phase happens in 4 different phases, that is prophase, metaphase, anaphase and telophase. For these, a different set of proteins are there which I am not going to discuss about now. Probably in the next video I shall do it for you. Now between the M and the G1 phase, we need another set of proteins which are called as cyclin B and CDK1, which makes the cell ready for synthesis. Having said all this, all these proteins cannot appear at the same time. Only the cyclins and the cyclin dependent kinases which are responsible for a particular phase of the cell cycle should be present. That means there should be a mechanism to stop the cyclins and cyclin dependent kinases which are not during that particular phase of the cell cycle. So we have what are called as inhibitors. The first inhibitor I have already shown to you, which is P16, which can block cyclin D once their job is done. Cyclin dependent kinase 2 inhibitors are present to block the function of cyclin A and cyclin E, and we also have additional blockers or protein arresters which can be P21 and P27. P for protein and 21 is the molecular weight of the protein, so it means the protein weighs about 21 kilodaltons and this protein weighs about 27 kilodaltons. The similar kind of mechanism can happen for CDK1, which can be done by CDK1 inhibitors. Cyclin B also can be arrested or blocked by a protein or a gene which is called as P21 and P53. P53 is another important enzyme which is responsible for DNA damage and cell cycle arrest. For DNA damage, identification and cell cycle arrest. So if there is a damage in the DNA, the cell will arrest, rectify the damage and then progress. But if the cell tries to evade this checkpoint mechanism, it can go on with the mistake and become cancerous. I shall talk about the cell cycle and the dysregulated cell cycle in the next video, in which I will try to explain more about cancer and how these cell cycle checkpoints play a very vital role in maintaining the proper divisional process of the cell without a mistake. Thank you for listening.